Hello everyone, my name is Arnav and today we'll be doing the first virtual challenge meet from 2023 to 2024 of the UIL science exam. I'll be going over the biology section in this video. We'll be going over all 20 questions, and before I get started with the 20 questions, I would like for you all to try the test on your own before I go over them. Right here I have the questions from numbers 1 through number 8. Here's numbers 9 through number 17. And here's number 18 to number 20. Now I'll get started on the questions in this section of the exam. Number 1. What term best describes corn that carries a bacterial gene that codes for an insecticide? So we have some corn, and then one of the genes codes for is altered, genetically altered, to code for an insecticide. Let's first narrow our search and eliminate the most obvious answers. B, transformation, is when bacteria takes DNA from another bacteria. So two bacteria share DNA with like one bacteria transfers its DNA into another bacteria. So the other bacteria will get that DNA. That's not what's being talked about, as this is between corn and a bacterial gene. Transformation is immediately out. Transduction is similar to transformation, except it's between a virus and a bacteria. So the virus injects its DNA into the bacteria. This is also not what's being talked about, so we can immediately eliminate answer choice C. Now, let's look at DNA. A is very tempting, genetically modified organism. D is also tempting, transgenic. Let's go over and define the two. A GMO is when an organism with its sequences altered, with its nucleotide sequences altered, or changed, or replaced, or deleted. GMO is very broad category. There's many, like, you can either alter, it could be an genetic deletion. There's insertion. There's so many different forms of like GMO. So it is broad. It's very broad. But it is support the answer. What about transgenic? Transgenic definition of transgenic is when you take the gene of one organism, put it into another organism. So organism 1, you take the gene, a scientist would take this gene, I'll color code it red, and you put that into another organism. So you put the same gene into another organism. This was the bacteria. This was the corn. Transgenic best fits the definition of what is being talked about. GMO, honestly, the reason why GMO is wrong is because it's way too broad. There's so many different ways organisms can be modified, and transgenic is way more specific than GMO. So the answer to number 1 is D. Number 2. What process takes place by reading the code of an mRNA strand? So, let's go over each and every process here. Replication is when DNA starts to extend. So you see the double helix DNA strand start to extend. So I'll label the double helix DNA strands. They just keep extending. It keeps going on and on. But this is talking about mRNA, not DNA, and breeding the mRNA. So A is completely out. Translation is the process when you read an mRNA strand to form a protein. It happens in a ribosome. An mRNA strand, I'll just draw the nucleotides, is read, and then amino acids come out of it. That is directly what the question's talking about. So B would be the right answer. Let's go over the other two. Transcription is the creation of an mRNA strand. So you have a DNA template. So let's say that these are the two DNA strands. One is a template DNA. The other is the non-coding strand. And the mRNA is formed on the non-coding strand. The non-coding strand. Remember. It's formed on the non-coding because it's a copy of the coding strand. So this is a simple drawing of transcription when mRNA is being formed from DNA. So this is the question's asking to read an RNA strand, not create one. So it's not that. Transversion is when you're turning a purine into a pyrimidine or a pyrimidine into a purine. So that means the purines are guanine and adenine. The pyrimidines are cytosine and thymine. Transversion is when adenine becomes thymine, or thymine becomes adenine, or thymine becomes guanine, or essentially when a purine becomes a pyrimidine, or a pyrimidine becomes a purine. That's not what's being talked about either, so the answer will be B for number 2. Number 3, glycolysis is considered a what process? So, we know glycolysis in cellular respiration. All you need is glucose, so you just need glucose, and the output are 2 pyruvates plus NADH plus ATP, 2 ATP, 2 net ATP. But here's the thing. Glycolysis, nowhere in glycolysis is oxygen needed for glycolysis. Oxygen is not needed as a reactant, and in fact, oxygen is only needed during the Krebs cycle. Krebs cycle is the first stage in cellular respiration where oxygen is needed. Not directly, but oxygen has to be there to start the Krebs cycle. So glycolysis is the only process in cellular respiration that's anaerobic. And the other 3 just are not the right answer because glycolysis is not aerobic. Number 4, the movement of molecules down their concentration gradient is known as what? Down their concentration gradient, that means from high concentration to low concentration in a container. Down the concentration gradient. That is an example of diffusion. Diffusion is from high to low concentration. And active transport is the other way around. Active transport is from low to high concentration, which is not what's being described. Bulk transport is when large materials are being transported in and out of the cell, which also is not what's being described. It's not talking about endo or exocytosis. And symport is when two molecules are going down in the same direction. So this is O2, this is H2O, let's just say. They're both going inside the cell, or they're going outside the cell. That would be an example of symport, but that's not what's being described. So the answer to number 4 is going to be B. Number 5. The type of covalent bond between the 2 molecules is called what? Looking at the functional groups of these molecules, you see that it has an amino group and it has a carboxylic acid group. And we know this is similar, this structure closely resembles what's called an amino acid. Amino acids consist of a central carbon, an amino group, carboxylic acid group, 1 hydrogen at the top, and a specific R group at the bottom. And bonds between 2 amino acids are known as peptide bonds. So they are known as peptide bonds. Glycosidic bonds are between glycogen strands, which are carbohydrates. That's not what's being talked about. Hydrogen bonds are bonds between hydrogen and another atom, most likely a non-metal atom like oxygen. That's also not what's being described. I have not heard of a nitrogen carboxylic bond, but nitrogen is not what's being bonded. It's in a whole amino group, so it can't be D. The answer to number 5 is A. Number 6. What is a possible characteristic that could be found in the identified part of the cladogram? So this X here marks where the characteristic is, marks the characteristic when it first emerges. So to study a cladogram, we know that on the left side, it's the old, like it's all the way in the past. So I'll actually, instead of writing old, I'll write past. And all the way to the right is the present. Or more modern, I'd say. So this trait emerged after the leuca was formed, after it branched to bacteria, after it branched to archaea. It formed way over here after all these are branched. If we draw a line from the tray and everywhere, all the species that branch off this red line will have this trait. But we know that eukaryotes are what branch off. It says eukarya. So we know this trait must be in eukaryotes, and it cannot be in any of these 3. It cannot be in bacteria, archaea, or leuca. So now we need to know what kind of trait is found only in eukaryotes. So let's look at all the answer choices. Circular chromosome is the exact opposite of what we're trying to find. Circular chromosomes are found in prokaryotes, and they're not found in eukaryotes. We're trying to find what's found in eukaryotes and not in prokaryotes. Nuclear envelopes are one of the main distinguishing factors between a prokaryote and a eukaryote. Eukaryotes have them, prokaryotes don't. B would be the right answer. Both eukaryotes and prokaryotes have multiple RNA polymerases, so C is not the answer. And prokaryotes, you'll learn about them later on, gram-negative bacteria and gram-positive, they both have peptidoglycan cell walls. So, and they're both prokaryotes, so D would be the wrong answer. So the answer, number 6, is going to be B. Number 7, correctly identify the structure in the image below. The only way to get this question right is just understand this, just memorize kind of like the structure. The answer to number 7 would be cholesterol, and the way we can determine this is we know that this is in the phospholipid bilayer. Cholesterol is found in the phospholipid bilayer, and cholesterol is one of the few lipids that have this sort of hexagonal shape right here. Since cholesterol is in the molecules in the phospholipid bilayer, and it has this shape, the only option for number 7 would be cholesterol. Number 8, animal phyla have distinguishing characteristics of germ layers. Which layers form first? If you guys have learned about germ layers, there are 3 different types. Hold on, we'll draw that a bit bigger. There are 3 different types. The germ layer in the middle is called, the first germ layer formed is called the endoderm. The second layer is called the mesoderm. And the outermost germ layer is called the ectoderm. All 3 of these germ layers form different features of your bodies. For example, the ectoderm forms skin. The mesoderm forms our lungs. And the endoderm forms more nervous tissue. So the first germ layer formed would be the endoderm. So the answer to this one would be letter A. Number 9. Which high-energy phosphate in ATP is removed first to release energy? If you guys remember the structure of ATP, there's 3 phosphates. There's a ribose sugar, a ribose pentose sugar, and there's adenine. The 3 phosphates from left to right are gamma. So I'll just write the gamma symbol. There's beta, and then there's alpha. So the outermost one's going to be released first. So number 9, the answer is C, gamma. So the gamma phosphate is what's going to be removed first. Number 10. A form of RNA processing includes all of the following except what? RNA processing is when you have a long, unaltered strand of mRNA and turn it into a much more concise and processed form of mRNA. There are 3 things that are needed for this. First, a normal RNA strand will not just be RNA, one big strand. There's going to be sections of it called exons, sections of it called introns. So let's call this exon. Let's call this one intron. The blue is introns, red is exons. Introns are parts of the mRNA that are just going to be cropped out by molecules called spliceosomes. Spliceosomes play a part in RNA processing by getting rid of the introns. So you'll only have one long strand of the exons combined. Then there's going to be something called a cap added to this. Make that green actually. There's going to be a cap added to the 5' end. This is for safety measures. This is just so that the mRNA can exit out the nucleus safely. So 5' cap is another part of RNA synthesis. There's also going to be multiple adenines at the very end of this, at the very tail of this mRNA. Again, for protection of the mRNA so that it doesn't degrade when it's out of the nucleus. The only process, or the only thing that's not involved in RNA processing is proteasomes, which are used to degrade proteins using a chemical called ubiquitin. It's not related to RNA processing at all, so D would be the answer to number 10. Number 12. What would be an example of intersexual selection? So what is intersexual selection? Intersexual selection is when one gender has a preference towards another gender, a preference of specific characteristics from another gender. So let's just say, I'll give an example. Let's just say a fish. A female fish prefers male fish with stripes. So male fish with stripes have higher reproductive success than male fish with, let's just say, dots. Let's say instead of dots, the dotted ones do not have a preference. The striped ones do. They have a certain preference for certain for certain traits of the opposite gender. So, which of the following describe that? Male elephant seals compete for the mating rights of a group of females. When members of one gender compete with each other to win the affection of another gender, that's called intrasexual selection. This A makes a difference. So A is actually not intersexual selection. Letter B. The female hanging fly will not mate with the male unless he brings her a large food offering. That means the male has to do something to win the female's affection. It's not really too close to my earlier example with the fish, but B is also another example of intersexual selection because the male has a certain characteristic of offering food that wins the female's affection. Letter C. Male lions have a larger mane while female lions do not. This is just a difference between males and females in the lions, so that's not selection. And male chimpanzees do not have a social group will often kill infant chimpanzees in hopes to take over as the head male. That is also that's not being described as intersexual selection because it's only talking about one gender. Number 13. The creation of a chromosomal tetrad forms during the process known as what? So what exactly is a chromosomal tetrad? A chromosomal tetrad is when two homologous chromosomes, so equal size and same genes, during meiosis 1, or actually I'll specify as prophase 1, during prophase 1 of meiosis, they attach to each other to form this structure called a tetrad. This is called the tetrad. So during this process, they exchange DNA. So after prophase 1, they're going to look something a little bit like this. But this one would have black gene, this one would have a blue gene. Something like that. They exchange DNA. So these two had just exchanged between each other. So what is this talking about? Where is this formed? Translocation is when it's chromosomal mutation. It's not defining this at all. Move this away. Yeah, sorry for that. Translocation is when one chromosome switches places with another chromosome. So you have one chromosome like this, another chromosome like this, and then segments of this chromosome switch place with, fill them out. Segments of this chromosome switch place with this chromosome. So the end result would look something like this, with most black but some blue. This is a chromosomal mutation that's not being described in this problem. This is an example of translocation, and that is not what's being described in the problem. It's not a chromosomal mutation between homologous chromosomes. This is a different process. So A is not the answer. Letter B. Interkinesis is essentially interphase 2 of meiosis. So that's also not what's being described. These two are very close to each other, but there's a key difference. Synapsis is the process where the tetrad is formed. Synapsis is this. This is synapsis, where the process where the two homologous chromosomes attach. Crossing over is more like this. This is more of crossing over. This is crossover. This is synapsis. Crossing over is just when DNA genetic materials exchange, while synapsis is where the DNA actually attached to each other. The chromosomes attached to each other. So the answer to number 13 is going to be C. Number 14. What would be true of the karyotype image below? We know this karyotype, it's definitely abnormal because in a human, there are 23 pairs of chromosomes. So 46 in total. This human has only 45 chromosomes. It's missing a pair in the 23rd pair. And we know the 23rd pair determines the gender. It should either have XX if you're a girl or XY if you're a guy. So it's definitely abnormal, so we can immediately eliminate A and B because it's missing one chromosome. And we know if you're missing a Y chromosome, this would be the X chromosome. It's missing a Y. There is no Y chromosome there. It doesn't matter, like, the X chromosome is not what's determining. It's the second chromosome that determines the gender mainly. If it's a Y, it's a guy. If it's an X, it's a girl. Since there is no Y, the main determinant is actually this. If there's no Y, it's thought to be a girl. So since there is no Y chromosome here, the answer for this one would be a female. Because Y determines the guy. It's just a small saying if you want it for this problem. Number 15. Isolated episodes of rapid speciation that occur between long periods of little to no change is what evolutionary process? So let's define all three of them. Let's start with B. Gradualism is just slow evolution over time, just very slow evolution. So just slow evolution over time. Now, is that what's being described? Here it says that there's lots of speciation. Speciation is lots of growth, lots of evolution between long periods of little to no evolution. That's gradualism is not what's being described here because gradualism, it's just slow over time. There's no rapid speciation in gradualism. Adaptive radiation is when stronger traits spread throughout a different population and more just adaptive traits and different forms of evolution spread throughout a population. That's also not what's being described in this problem. Natural selection is when the fittest, or the quote-unquote fittest, because it's not just physically, the organisms with the best traits in their environment to survive would have a higher chance to survive and reproduce, to pass on these traits to their progeny. That's also not what's being described. Letter A. Punctuated equilibrium is rapid speciation, so rapid evolution, between no growth, between periods when there's no growth at all, little to no growth. So, number 15, the answer choice would be A. Number 16. Which of the following descriptions is true of mitosis? Mitosis is when a somatic body cell, so somatic cell, also known as a body cell, so your normal cell, skin cell, blood cells, muscle cell, a somatic cell with 2 N chromosomes divide to make daughter cells with 2 N chromosomes. Now, we can immediately eliminate B because it does not create haploid cells. Haploid cells would be N. N would mean haploid. 2N would mean diploid. So it cannot create haploid from diploid. That is actually an example of meiosis. Letter A. Only occurs in germ cells. That's not true. Normal body cells can also undergo mitosis. And letter C is also talking about meiosis, requiring homologous chromosomes to bind together. Just a fun fact, this is kind of describing number 13, which we did earlier. But letters B and C describe meiosis. A non-reductive process is when the number of chromosomes do not change. So initially it was 2 N chromosomes. At the end it's 2 N chromosomes. So the number of chromosomes stayed the same. The answer to number 16 is going to be D. Number 17. During photosynthesis, where do protons begin to build up as a byproduct of water being broken? So, we know this process is we're given that it's photolysis. Photolysis is a process that happens during photosynthesis. And we know photosynthesis takes place in chloroplasts. So if you guys have no clue what like photolysis is, if you're not having gone that deep in honors bio, you can immediately take an educated guess right here because you know it has to happen in the chloroplast. Photolysis is just is absorbing light. So you know that it's during the light reaction of the chloroplast. B and C do not even have anything to do with the chloroplast. Cytoplasm is located outside the chloroplast. Mitochondrial matrix is located in the mitochondria. So you can eliminate B and C and take a guess between A and D if you don't know. Photolysis, I'll diagram the process of. This is not the chloroplast. This is something called a thylakoid. Thylakoids are these like green pancakes like structures found in the chloroplast. They're the ones that actually where the light reactions take place. So I'll write light reactions. You might cover this in honors bio. So we know water is broken down by photolysis. So there's going to be because water is broken down, it's broken down to 2 hydrogen and 1 oxygen. The oxygen binds with another oxygen to make a molecule called O2 that leaves the thylakoid. So now you're just going to keep building up hydrogen molecules. There's going to be a huge build of hydrogen molecules inside the thylakoid. So the answer to this one would be the thylakoid. D, the thylakoid lumen, which lumen is just a specific word to describe the inside of the thylakoid. And the stroma is essentially the liquid outside the thylakoid. This is where the light-independent reactions take place, so it's not related to photolysis. Number 18. How does RNA differ from DNA? Let's go over each and every one. Let's go over the wrong answers first. RNA nucleotides are bound by peptide bonds. If you remember earlier, I told you that peptide bonds are between amino acids, so they form proteins. Peptide bonds form proteins. Both of these are called nucleic acids. RNA and DNA are nucleic acids. So you can immediately eliminate answer choice D. That's just not right. RNA is a double helix. All the forms of RNA are all single helixes. mRNA, tRNA, and rRNA are all three of them are single helixes. And RNA includes nitrogenous bases: adenine, thymine, guanine, cytosine. RNA does not contain thymine. RNA contains a nitrogenous base called uracil instead of thymine. It does contain the other three, though. So D is the incorrect answer. A. RNA ribose has a hydroxyl on both the 2' and 3' carbons. The main difference between RNA and DNA, it's actually in the name. DNA stands for deoxyribonucleic acid. That means the sugar only has a hydrogen here. There is no oxygen. That's why it's called deoxy. Deoxy means no oxygen. This is the DNA sugar. The RNA sugar, on the other hand, there's one here too. The RNA sugar, on the other hand, does have oxygen. It has oxygen binding with hydrogen. Oxygen binding with the hydrogen. That's why it's called ribonucleic acid instead of deoxyribonucleic acid, because it has oxygen in it, so it's not losing. It doesn't have a lack of oxygen. So the answer to number 18 would be A. The process of ammonification occurs by bacteria in the soil and the conversion of what to what. So, what exactly is ammonification? Ammonification is a process in the nitrogen cycle, which I'll be going over. So this would process in the nitrogen cycle. So, the nitrogen cycle, I'll actually quickly diagram the nitrogen cycle before I go over what ammonification is. Nitrogen cycle starts off as nitrogen gas in the atmosphere. The nitrogen gas in the atmosphere, due to instances like lightning, for example, like from a lightning cloud, due to that, the nitrogen gas gets sent to the soil and is converted into a compound called ammonia. with the help of bacteria too. There's bacteria called nitrogen-fixing bacteria that turns nitrogen gas into ammonia. This process of nitrogen gas into ammonia is called nitrogen fixation. Nitrogen fixation. Now, ammonia can enter a plant called assimilation, in a process called assimilation, or it can become a compound called nitrate. It can become a compound called nitrate, NO3, in a process called nitrification. Let's write it over here. In a process called nitrification, ammonia can become nitrates. So, after nitrates have two processes they can go through while in the nitrogen cycle. They can either assimilate into plants, just like how ammonia does. Either they can do assimilation, or they can undergo a process called denitrification, which is when they just leave the atmosphere. This process is called denitrification, where they end up becoming nitrogen gas all back together again. Now, when animals die, when this animal died, his amine compounds were added into the soil to create a chemical called ammonium. This chemical is called ammonium. This process, when the dead animal compounds became ammonium, this process is called ammonification. Ammonification. And once the ammonium then gets converted into a compound called nitrite, NO2, nitrite. This ammonium is converted to nitrite, which is then later, by bacteria, is then converted into nitrate, which we discussed earlier. Write the name, nitrate. And then the whole cycle repeats itself again. So this is the basic diagram of the nitrogen cycle. Now, what is the question asking? The process of ammonification occurs by bacteria in the soil, and the conversion of what to what. So let me highlight just the key words you want to know. Ammonium, there. And then nitrogen gas. Okay, now back to the question. The process of ammonification occurs by bacteria in the soil and the conversion of what to what. Ammonification is a conversion of the dead animal to the ammonium. So dead animal to ammonium. Which answer choice talks about that? C talks about amine compounds from a decaying organism into ammonium. So answer choice C would be the right answer. Now, what about the other options? What are they talking about? For answer choice A, nitrogen gas into ammonium, that's talking about nitrogen fixation. That's not talking about ammonification. That's talking about this process all the way over here. So A is the wrong answer. Nitrates into ammonium. That's not a thing. Nitrates don't become ammonium. Ammonium becomes nitrite. That becomes nitrate. So B is not the right answer. And ammonia to ammonium, that's not the right answer either. Ammonium does not become ammonia. So D is the wrong answer. So the correct answer to number 19 is going to be letter C. Last question, number 20. Guard cells in a plant leaf structure are responsible for what? Guard cells are responsible for letting oxygen out of the plant and carbon dioxide in the plant. So guard cells are responsible for opening and closing certain holes called stomata. Stomata let oxygen out of the plant and carbon dioxide into the plant. So the guard cell's main job is just to open and close the stomata. So the answer to number 19 is going to be letter B. There's no molecule that opens and closes the xylem and the phloem, so A is not really an answer choice. Letter C is talking about the xylem. Xylem transports water throughout a vein. And answer choice D is talking about the phloem. The phloem. Okay, I keep pronouncing it wrong. The phloem. Phloem transports sugars throughout the plant. So transporting sugar, moving water, xylem, phloem, which are not the answer. It's not what's being described in the question. So the answer to number 20 is going to be B. All right. We're done with the 20 questions in the biology section of the virtual challenge from number 1. If you guys have any questions regarding any of these 20 problems, make sure to let me know down in the comments. Feel free to subscribe and like the video if you enjoyed or if you found me helpful in solving these problems. I'll be seeing you guys when I post my next video.